Cool. So do you think we can start? Or is there no one else joining? I guess should be fine. Cool. So basically today, I would like to tell you a bit about molecular genetics, about what you need to know, but more importantly, why molecular genetics is by far the most exciting field of medicine. And I would also like to encourage you to potentially think about your FHS in the future. So if you have any questions about FHS, PhDs, or anything, just let me know. So you can either email me or ask at the end. And also just interrupt me at any point if you have any questions. Okay, cool. So as I mentioned, I would like to briefly go through your syllabus today. So there is quite a lot to cover, so I'm not even trying to cover everything, but I will try to cover most of the most important points and come up with some mnemonics or some ways for you to remember the most important things, especially for the MCQs. Yeah, and I will try also to give you some understanding of the entire field. So then you can easily just expand your knowledge if you're more interested. Yeah, and once again, if anything is unclear or you want to learn more about anything, just let me know. Yeah, so as you will see here, there is quite a lot of material. It starts from just overall overview of the genome, and with then quite a lot of focus on different diseases and mutations. So I will try to cover it. I will not really cover cancer genetics because I saw this is covered in another lecture. But after a quick introduction, I will focus on the methods because I thought methods are kind of most important for you to really understand the field and potentially write your essays if you want at the end of the year. And then we'll also go through all the most important points. Cool. So just to start with, I would like to just tell you my idea of molecular genetics and how I interpret it. So for me, is there something new in the chat? No,
Sorry,
just
that's just me inserting
yeah,
the.
no worries. So how I imagine the genome is like one big library with 23 pairs of books, which are quite massive books because each of these books has between 60 million and 250 million pages. And what's quite interesting is that most of these pages are blank. So only around 1 to 2% of these pages are actually filled in. So that's exactly what happens in the human genome. So we have 23 chromosomes that vary from 62 to 150 million base pairs, together forming around 3 billion base pairs of the genome. And you might have heard about this junk DNA hypothesis, which was quite popular, stating that this big majority, this 98 to 99% of the genome is just useless because it's not coding for anything, it's not really transcribed. But over the last years, people have rejected this hypothesis because it's more and more clear that these non-coding fragments of the genome are actually really important for different processes, including DNA protection and DNA segregation, but also the regulation of the transcription of the genes. Yeah, so as I mentioned, only around 1 to 2% of the genome is actually coding something, and half of it will be coding genes. So we have around 20,000 protein coding genes in the genome, and around the same number of non-protein coding genes, which will be those coding, for example, long non-coding RNAs or other types of RNAs that don't lead to protein formation, but instead usually just regulate the transcription of the coding genes. Okay, so what's the structure of the DNA? So starting from the very basics, DNA is built of purines and pyrimidines, so adenosines, thymidines, guanines, and cytosines. And I always struggled a bit to remember which ones are which. So hopefully after these short videos, you will actually remember. So if you think about this pyramid made of the
Go
Twig
dip with a new
crackers,
chili meal twist. Enjoy Duck and Dip.
you might hopefully remember that the pyrimidines are thymidines, uracil, and cytosine. So that's basically how I remember it. And if you also think about these holes, there will be lots of them, but all the holes are single. And this is basically how I remember that pyrimidines consist basically of a single ring, while purines will have a double ring, which might be an important thing to remember for MCQs potentially. Okay, so what's the structure of the DNA? So on the most basic level, you will find those four nucleotides, A, T, C, and G, that consist of the base, which is kind of the most important. But also there will be sugar, so either deoxyribose in DNA or ribose in RNA, and the phosphate group. And this phosphate group is quite critical for joining the two nucleotides together. So this is how they join covalently. On the other hand, the bases, the nitrogenous bases join together with the hydrogen bonds. And one important thing to remember is that adenosines join thymines with a double bond, while C joins G with a triple hydrogen bond, which is why the GC bonds will be stronger, and it will form more, and they will form stronger, more compact chromatin. Okay, so having a very basic overview of what DNA is and the organization of the genome, I would like to tell you something about the key techniques we are using in studying DNA, in studying the genome. So I will start from the most basic ones, so the sequencing methods. And the first one that you will have likely heard about is the Sanger sequencing, which was the first technique developed in 1970s. And I think in 1980, Fred Sanger from the LMB in Cambridge got Nobel Prize for it. And it is very similar to how we replicate our DNA. So you put a template, you put a primer that binds somewhere within this template, then you start the replication of the DNA. However, except for the normal DNTPs, which are just nucleotides that get incorporated, you also get D-deoxy nucleotides. And if you incorporate such a nucleotide, the replication terminates. So you can't build any more nucleotides to it. And on top of that, these D-deoxynucleotides are fluorescent. So each of them has a different tag attached to it, so a different color of the tag. So you run lots of reactions like this. So this get incorporated randomly. So the replication terminates randomly at different stages. And so then you get these fragments of very different lengths. You run them on the gel. And what happens is that the shortest fragments will migrate faster than the longer ones. And this is how you basically see that the longer fragments, for example, will have G attached, while the shorter one will have T attached to it. So just to show you the real data, so when you send your sample for sequencing, you get a result like this. This is one of the samples I sent like a year ago, and you see a peak for each basically position in your DNA. You see a peak of a different color, and the software interprets it for you as different bases. So this is the most basic sequencing method. At the moment, it's not used for sequencing entire genomes, but it's used as a very cheap method to sequence very short fragments. So usually up to 1,000 base pairs. You can do it basically for like 3 or 4 pounds per sample, so it's pretty cheap. And interestingly, it's also the basis of the Human Genome Project. So you will probably have heard of the Human Genome Project, which is a project started in 1990, mostly in the US, founded by NIH, but also Wellcome Trust here, which cost around $3 billion. And the researchers used mostly Sanger sequencing to sequence very short fragments of the DNA from the genome, then to assemble it together to then get the entire sequence. So this 3 billion base pairs. So then the paper came out claiming that it's the first human genome assembly. So now we know the sequence, and since then we got another maybe 10 papers saying exactly the same, because they covered only around 92% of the entire genome, but they didn't cover the sequences that are difficult to sequence. And basically, if you're interested in it, you can have a look at this paper here and that telomere to telomere assembly 2 years ago, where they, for the first time, claimed that they sequenced everything, which is not entirely true, but is really close at this point. And obviously, they used very different techniques, and I will talk about some other techniques that they used in the next slides. Okay, so another key method for sequencing that you need to know, because it's used everywhere in the NHS, in research, everywhere, is the Illumina sequencing, also called new generation sequencing. And it's in some ways a very primitive technique, because you just get genomic DNA, you fragment it into small pieces, usually 50 to 200 base pairs, so extremely short. You ligate, so you attach adapters on both sides, and these adapters will stick to the membrane. And after they stick to the membrane, you add nucleotides that have different dyes attached to them. So each nucleotide will emit different light. And then polymerase arrives and basically just builds this complementary strand of the DNA. And whilst doing it, you see these different colors of nucleotides attaching one by one. And by doing this, you can actually sequence entire genomes. So this is an extremely important innovation developed mostly by Illumina in Cambridge, because it actually allowed us to upscale our experiments, and now sequencing of the entire genome can cost as little as, let's say, £600, depending how many reads you want. And this is being done currently routinely on the NHS. So the advantage is that you can sequence a lot, you can do it quickly, but the disadvantage is that these reads will be extremely short, so 50 to 200 base pairs. So if you want to assemble a new genome where you want an overlap, it's going to be difficult. And the same happens with the repeats. So big chunk of the genome consists of the repeats. So sometimes 3 nucleotides keep repeating many times, sometimes longer fragments keep repeating. But the challenge with such short reads is that you can't really tell how many repeats you've got, where exactly you map. And this is why we needed more methods to study DNA. And I think the most important method that you need to know about is nanopore sequencing, which was developed here in Oxford and is the second most widely used method for sequencing DNA currently. So I used it quite a lot in the lab, and what's quite incredible is that you can use such a small device that is just connected to your computer. You prepare the libraries, so you prepare DNA before on the bench, and then you just inject it, and the sequencing is running on your computer. So it's really portable, but its biggest advantage is that you can sequence a lot longer fragments of DNA. So my longest read was over 1 million base pairs, which is unusual, but you can easily get thousands of base pairs per read. So these longer reads actually allow you to assemble genomes a lot more easily and look for different transfers of the DNA or multiple repeats. So how does it work? It's actually got a very different mechanism, because a nanopore consists of this membrane and around 512 pores in each of the flow cells. And each of these pores contains a motor protein, which basically pulls one strand of the DNA through it. So if you imagine these are different nucleotides, and this will be, let's say, ATCG, each of them will change the current across the membrane in a different way. And this is how nanopore interprets what nucleotide just passed through it. So I hope you can appreciate how smart is this technology, but also how unreliable it can be. So nanopore sequencing is characterized by poorer quality of reads, and it's also more expensive, quite a lot more expensive to sequence a lot. So it is important in some areas of research, especially as, for example, I was sequencing not just ATCG, but also other artificial nucleotides by it. But then it's not used commonly in the healthcare. There are many ideas how to incorporate it into designing better treatments, but it's not yet in the NHS. Yeah, the last one, if you're interested, will be PacBio, but just for the interest of time, I'm gonna skip on it. Okay, so these are the key methods for sequencing the DNA. I would like to also show you some methods for studying other aspects of the DNA. So not just its sequence, but also its positioning and the proteins that attach to it. So the first one that I think is one of the prettiest one will be FISH. And FISH is based on metaphase spreads. So that's a metaphase spread. So here I just arrested cells basically in the M phase of mitosis. So basically right when they are just about to divide. So all the chromosomes are condensed, as you can see. And then you drop these cells from around 50 centimeters or a meter on top of a slide. And this causes these chromosomes to actually spread nicely. And then you stain them with, for example, DAPI, which is probably the most widely used DNA dye that binds to them. And you can see something like this. So here you can see not only chromosomes, quite a lot of them because it's a colon cancer cell line, but also a number of smaller structures that are called extrachromosomal DNA. And they are extremely powerful for around 1 in 6 of all cancers because they carry a lot of oncogenes, so the genes that drive cancer. Yeah, today we don't really have time to talk about them, but if you have any questions, you can ask me later. And I would encourage you just to read around them. Okay, so that's metaphase spread. And what you can see here is FISH, so fluorescent in situ hybridization. So it's a very similar method to metaphase spread. So you do exactly the same thing. You drop your mitotic chromosomes or entire cells, and then you add short DNA fragments that are labeled with fluorescent dyes. So here there will be one sequence binding to chromosome 8 that is labeled in green, and one binding to CMYC, which is a key oncogene, labeled in red. And what you can see here is that this and this will be chromosomes 8. But what's really extraordinary is that you can see this much of CMYC, because usually you see basically just one copy, so one small dot. But what happened in this cancer is that there was a massive amplification. So basically there's an entire chromosome consisting of the CMYC, which is driving this cancer to be more aggressive. So if you remember one oncogene, it could be CMYC, which is an extremely powerful one. Okay, so we also use it to look at different abnormalities, genetic abnormalities in cells. So for example, here you can see Philadelphia chromosome, which is a translocation between chromosome 22 and chromosome 9. So basically the ends of chromosome of these chromosomes join together. So ABL1 gene, which is another oncogene, joins the promoter of highly transcribed BCR gene. And this way it's extremely highly transcribed and is driving tumor progression. And this is a very frequent translocation in CML, which is chronic myeloid leukemia. So if you're interested in leukemias or want one clinical example, yeah, you can think about it. Okay, so In the previous slides, I showed you how we look and how we look for the different translocations of different fragments of the DNA in the genome. And now I would like to show you how we look for how many copy numbers of the genes we have. So for this, we use PCR, which is a standard technique that many of you will have used already in the lab, where you can amplify the DNA. So you have one copy of the DNA. In each subsequent cycle, you get 2 copies, then 4 copies. So you basically double the amount of the DNA that you have. And the process is quite simple. So you take your double-stranded DNA, you put it in 95 degrees, so the 2 strands separate. Then you go down in temperature to around 68 degrees, so the primers attach, because primers are necessary for the replication to start. And then in around 72 degrees, polymerase attaches and starts replicating those fragments. So in each cycle, you get twice, in each subsequent cycle, you get twice as much DNA as in the previous one. And it's a method extremely often used both in research and in diagnostics. So with a very small modification, you can also use it to detect how many copies of a gene you have. So for instance, here, I put this graph because you can often find it mostly in year 3 on the paper analysis, but yeah, it's also quite common to have to interpret such a graph where you can see different samples labeled in different colors. And on y-axis here, you can see the fluorescent levels. So then you set up a threshold, let's say here at 3, and you look at which cycle does your sample hit this threshold. And you compare it between the different samples. So for example, if you compare green with orange, you will see that green hits this threshold at cycle 17, let's say, while orange hits this threshold at cycle 20. So there is a difference of 3 cycles. And this means that the difference between these samples will be 2 to the third of our specific target. So here we'll have 8 times, the green sample will have 8 times as much of this gene as the orange sample. Yeah, so it can still be used quite a lot for detecting the copy number variations of specific genes. Yeah, and we use this PCR method as well as other methods for molecular cloning. It's also one of the techniques you need to know according to your syllabus, and it's used a lot in research. In particular, like when you want to put a gene of interest to your cells. So for example, you want to mutate this gene, so you mutate it as you wish outside of the cells, and then you insert it, usually using transfection inside your cells, and you see how these cells change. So in order to do that, you need the backbone of a plasmid, which contains usually antibiotic resistance, so you can multiply it in bacteria and then select for it in the human cells, which you cut with restriction enzymes that cut very specific sequences of DNA. And then you basically ligate it with your gene of interest here in yellow. So you put this together, you add ligase, and then you get this plasmid. Then you transfect it into the cells, for instance, and they start producing the proteins that you want. So you can study how they behave. So if you ever do projects in genetics, you're very likely to do a lot of cloning. Yeah, and then if you have either such a plasmid or fragments of the DNA, you can separate them using DNA gel electrophoresis. So like I showed you before, each nucleotide has this phosphate group at the end. And this phosphate group gives DNA quite negative charge. And this is why the DNA will migrate from the negative to the positive electrode. And of course, the smaller fragments will migrate a lot faster in these agarose gels. And this is how you can separate shorter from longer fragments. And that's something you will do a lot in the lab, but you may get also questions about it in your exams. Okay, so I showed you some of the most important techniques that people use in the lab to study the organization of the chromosomes, but I haven't really talked about what else is there in the nucleus except for the DNA. So the naked DNA is quite rare because naked DNA is quite fragile and would take also a lot of space. So we need to wrap it around something to protect it, but also to condense it. So it will be wrapped around histones. So around 8 histones, 4 pairs of separate histones, H2A, H2B, H3, and H4, and histone H1 at the end to basically give a seal, to put a seal on this entire complex. So this around 150 base pairs of DNA will be wrapped around. To form this structure, this nucleosome array, which can then be further condensed with different proteins in cells. And the 2 terms that you may want to know is euchromatin and heterochromatin. So euchromatin will be a loose chromatin without that many potentially histones or with more relaxed histones that will be accessible to polymerase. So as a result, it will be transcribed a lot. And heterochromatin will be a very tight, compact DNA that often won't be transcribed almost at all, if not at all. So how does it really change? So there are two main ways of condensing or opening your DNA and regulating it in different cells. One of them will be DNA methylation, which I won't talk about too much, but mostly the GC-rich regions will be methylated, which will make them more compact and less transcribed. And another one will be histone modification. So like I told you, each nucleosome has basically 8 key histones, and each of the histones will have a C-terminal tail, so quite a long tail sticking out. And these tails can be modified either with methylation, acetylation, phosphorylation, or ubiquitination. So methylations are usually the most important, so different types of methylation will activate the transcription of the DNA, while others will make the DNA more compact and will suppress the methylation, suppress the transcription and protein production from the DNA. And similarly, for example, when you get DNA damage in specific region, you can get incorporation of a specific variant of histone H2A, which is gamma H2AX, which you will also find a lot in the research papers because it's one of the markers for us of the DNA damage. And how can we study it? Well, there are quite a lot of methods for it. One of them that you should know about will be Western blot, where you can collect your cells, you lyse them, so you lyse all the cell membranes and everything to release the proteins. Then you basically boil everything and you inject these proteins into different wells of SDS-PAGE gel. Then you put this gel in a big tank where, once again with the current, so the proteins migrate. Usually the big ones will migrate slowly, the small ones will migrate a lot faster. And then you transfer these proteins from the gel to the membrane. And on this membrane, you want then to detect that proteins. And you do it using antibodies. And you've probably heard about primary and secondary antibodies, where the primary antibodies are produced, for example, in mouse. So you can produce them in different animals, which is quite important. And this antibody will bind to our protein of interest. And then we add the secondary antibody, which will be produced in another species, for example, in rabbit, and it will bind to our mouse antibody. And these secondary antibodies will be linked to different fluorophores, so they will emit signal And this is how you detect your proteins then. And this is important for two main reasons. First, it's difficult to make primary antibodies with fluorophores that would be emitting signals, so it makes entire process a lot cheaper. And second, a lot of these secondary antibodies can attach to these primary antibodies, making the signal also a lot stronger. So just to give you an example of a Western blot, if you have to interpret it anywhere, so on the left you usually get a ladder, so unknown proteins that you buy that migrate at different heights. So you know that the lowest one will be at 20 kilodaltons, while the biggest one will be around 250. And then in your samples, in the right, you will be looking for your protein of interest that you basically look for using antibodies. So you can see that this protein exists quite a lot in the sample on the right, but is absent in the sample in the left. Yeah, so the main advantages of this method are that it's quite specific for the proteins you want, but then you need to know what proteins you're interested in. So it's not like mass spectrometry where you basically inject the sample and it tells you all the proteins that exist there. Here you need to have an idea what you're looking for. So that's quite a big limitation. And also sometimes it technically, for some proteins, it can be challenging. Okay, and I think the last method that you need to know is CRISPR-Cas9. You must have heard about it quite a lot recently after the Nobel Prize for Jennifer Doudna and Emmanuel Charpentier a few years ago. So it's the first method that revolutionized molecular biology research because since they discovered CRISPR-Cas9 from bacteria, you can basically cut the DNA wherever you want. The only important thing is that you get a PAM sequence quite close to your target where you want to cut, because it allows the Cas9 protein to bind to that region and cut exactly where you want. So then we use this CRISPR-Cas9 system quite a lot to either knock out genes, so just cut them and hope that they won't produce functioning proteins anymore, or for gene manipulations when you want to move fragments of the genes around. So you can really do a lot with it in different ways. Okay, so you must be quite tired. So if you want, we can do a quick session of Menti if it works out. So if you could just join this, join menti.com using this code 12770450. I removed it from the presentation because it didn't work before, but hopefully it should be okay now. So if you try to join it using this code, it should work out. But yeah, let me know if it doesn't work. Yeah, so let's give you another 30 seconds to join. Okay, brilliant. So here I just put very easy and basic questions for you, just about the most important things to remember for MCQs. So can you see the first? Yeah. Now you should see the first question. So you have 30 seconds to answer. Okay. Sorry, can we see the? Yeah. Okay. Yeah. So yeah, most of them remembered. Most of you remembered correctly. So yeah, if you remember the pyramid, hopefully you will remember for a long time. And that's something that often comes up in MCQs and will be also important for your research careers. Okay. So question 2. Let's move on. Oops. So here, just to see if you remember how we discussed different sequencing approaches. Okay, so we have quite different responses here. So, yeah, so it was a difficult one because, yeah, the keyword here is DNA repeat. So we are thinking about like repeats such as like when you're looking at Huntington's disease, for example, if you have hundreds of repeats, you won't be able to find out how many repeats you have if you have 50 base pairs reads from Illumina. So you need something longer. And this is why you basically need nanopore sequencing. With Sanger, you can try, but it's also struggling with dead repeats. And another thing I quickly mentioned to you before is that you can detect DNA methylation, so changes to the DNA, or this substitute nucleotides using the nanopore. So it's quite a fascinating technique. Okay, question 3. Okay, brilliant. Yeah, so Illumina won't be usually used for plasmid sequencing because, uh, yeah, it's quite expensive and a lot of work to prepare those samples. And then you get lots of reads, lots of short reads. And in fact, here you want few reads, but that are long ideally. So Sanger is the best because it's quick. I also put Nanopore as the correct answer because right now there's this service called Plasminosaurus where you send them your plasmids and they just run hundreds of plasmids in parallel all together on the same flow cell, and then they send you the results. So Nanopore can in theory work as well, but yes, Sanger is the right answer here. Great, so well done everyone. Are we done? No. Yeah, so what's best for whole genome sequencing when you want to sequence a lot, just like they do right now in this? Is it 500,000 babies project, basically, where they now sequence a lot of neonates? Yeah, so Illumina is the right answer. So they used Sanger in the initial Human Genome Project, but it's extremely inefficient and it cost $3 billion to assemble the genome. With Illumina, you can do it really quickly, really cheaply. You can basically sequence entire genome and you can find out mutations. Okay, and question 5. Okay, yeah, that's brilliant. So yeah, exactly, it's fish. It's these pretty images that I showed you where you can see uh, your sequences of interest put actually in space, either on chromosomes or in the interphasic cells that are not dividing. You can just see the dots in the blue nuclei. Okay, and the last question. Yeah, how to detect how many copies of a gene you have. So for example, you take tumor biopsy and you isolate DNA from it, and you want to see if your gene of interest is actually amplified or not. So you can select the right therapy. Perfect, so it was kind of tricky. Oh, sorry. It's my fault I didn't select the right answer, but yeah, it's qPCR. So with PCR, you just amplify the DNA, but you can't detect how many copies you have. With qPCR, you add either cyber, so either a dye to detect how much DNA you have, or a Tagman probe, which basically was used during COVID So Tagman probes bind to your proteins of interest and, sorry, to the DNA that you're interested in, such as the COVID DNA. And then when the polymerase basically replicates DNA, it cleaves this fluorophore. So then it starts to emit the light. So every cycle it emits more and more light. So this is how we used to detect if you have COVID in your body. Okay, so back to the slides. Let's get back to some basics. So what are mutations? I'm sure you all know that we can have different mutations in your DNA and different types of mutations. So they can have very different mechanisms. They can be either random, so just by replication, you sometimes introduce random mutations, or when you're exposed either to UV light, to the sun, or when you're exposed to when you smoke cigarettes, then you can have different patterns of mutation. And for example, when you smoke, you often get cytosine deamination, which then looks like uracil. So then it's recognized as uracil, so it binds adenosine. And then later it's basically, so adenine binds then thymidine. So this is why you get this switch from C to U. So C to T switch is a common one for smoking. So this is not important for you to know, but what's quite important, I feel, is to have this idea that we get different types of mutations, and we often can detect actually what caused them with some degree of certainty. Okay, so what are the most important types of mutations that you know about? If you're looking just at the point mutations, so changes to just one nucleotide, you can have silent mutations, missense, or nonsense. So silent ones will be, as you might expect, they don't change anything. So for example, here you change GTA. This A is changes to T. And both GTA and G2T code valine, so nothing changes. And as a rule of thumb, quite frequently the last nucleotide in the triplet, in the codon, is not as important. So quite often it doesn't change anything. Okay, so missense mutations will be, as you will expect, change the sense of the amino acids coded. So we'll change the amino acids. So for example, here from CCC, this C will be replaced by adenine. So to form ACC, so proline will transform into threonine. And finally, nonsense mutation will cause this stop. So if you're talking nonsense, it doesn't make any sense, so you don't produce anything if you think about it. So TAC, so tyrosine will change to TAG, which is one of the stop codons. So stop codon will will stop the translation, and you won't produce, you won't add any more amino acids, which will likely lead to a defective protein. Okay, so these are the key mutations on a single base level, where you just change one nucleotide. On the other hand, indels or deletions can insert multiple or delete multiple nucleotides. And what's quite important to understand is that if you remove or add 3 nucleotides, it doesn't change as much, because then you just remove or add a single or multiple amino acids, but often the protein can still be functioning. But what often happens is that you add or remove 1 or 2 nucleotides, or 4 or 5, which then leads to frame change. So if you look at for example, here at this example, you will see that that this mRNA will be translated up to here, and then if you add an additional nucleotide here, you used to have GAA, and currently you have GGA and AGA. So this and all the subsequent amino acids will be affected, so will be different. And usually at some point you also hit the stop codon, the premature stop codon, so the proteins will be completely messed up. So yeah, so this tend to be more catastrophic. And of course, especially if we think about cancer, one thing, if you're interested to read about, is chromotripsis, where basically entire chromosomes, for quite unknown reasons, just break into small pieces and then rejoin together. So they create a massive mess where all the DNA is rearranged, and you can create that circular extrachromosomal DNAs that I told you about before. Okay, so another MCQ. So I'll give you another, yeah, around 30 seconds just to try to think, try to match, yeah, the list on the left with the list on the right. Okay, so let's have a look. So yeah, so let's fit the right mutation type with the right description. So all amino acid sequence downstream of the mutation is altered. So it's gonna be frame shift. As we talked, if you add 1 or 2 or 4 or 5, just not 3 or 6 and so on, nucleotides, or you remove them, all the, like the way you read the DNA will shift and will produce very weird and non-functioning proteins. Okay, next one. No change occurs to the protein coding sequence. It's quite easy, silent. The protein produces shorter than normal. Nonsense, as we talked about it, because you hit the premature stop codon. The mutation that affects males, this we haven't talked about, but yeah, these are X-linked. And I believe you have another lecture where you will be talking about that. And the mutation resulting in the substitution is the missense mutation. Cool. So we have another 10 minutes, so I'll try to just cover as much as as we can, but you will have the rest of the information on the slides. But what's quite important for you to understand is that DNA replication in humans is semi-conservative, meaning that you have 2 strands of the DNA, and before the replication they will separate in the replication bubbles, and then there will be 2 polymerases, one attaching to one strand, the other to another strand, basically synthesizing DNA. And what it leads to is these 2 new double-stranded DNAs, and each of them will have one old and one new DNA strand, which is why it's called semi-conservative DNA replication. Yeah, and as you will know, probably already, it occurs during the S phase of the cell cycle with small exceptions. Yeah, and then they are being segregated between the daughter cells. Okay, so how does the replication initiate? So that's again something that is interesting to know, but you don't need to remember everything here. But what you need to know is that there are some key enzymes that are being involved. So for instance, DNA helicase, which untangles the DNA to form this replication bubble, as you can see here. But if you think about the twisted DNA and you think about one protein that untangles it to create, basically just stretches it to create that replication bubble where you can, when the polymerases can attach, the strands further on will twist, will either twist or untwist, depending also how you do it, but there will be likely a torsion and pressure on the DNA. And if you just kept doing it and pushing this replication bubble that is traveling throughout the DNA, it will push more and more DNA until it breaks, basically. So in order to prevent that, there are those enzymes DNA topoisomerases 1 and 2. Topoisomerase 1 will just cut one strand, topoisomerase 2 will cut two. And yeah, and as you can imagine, those topoisomerases are quite key targets in cancer treatment because cancers tend to replicate really fast. There will be the polymerase will proceed like crazy with very high speed. And there will be lots of them, creating lots of pressure on the DNA. And if you don't have these topoisomerases, this DNA will start breaking, leading often to cell death. Okay, and also DNA can't just start just like that from a random place. You need a primase, so an enzyme that will synthesize just a very short fragment of DNA, of RNA first. Then the DNA polymerase II joins and starts replication. And then this RNA is being replaced by DNA. Okay. Yes, and something we have talked about but haven't named it yet will be the Okazaki fragment, something you definitely need to know about. That is quite fascinating because as I showed you the replication bubble before, there will be leading and the lagging strands. So the leading strand, you will be basically replicating it, and the replication will be progressing here, let's say to the left. And the entire bubble will travel to the left. So what happens is that one strand will be replicated easily. You can just proceed continuously, but the other one, sorry, like basically it will go down and here it will finish. And you already opened this bubble further down on the left. So you need to generate, to start replication from here and then join these fragments. So these fragments are called Okazaki fragments. And if you have problems with understanding any of the techniques I showed you before, or how the replication works or anything, YouTube is usually your best friend because then you can actually see how it happens. And it's quite fascinating. Okay, so these Okazaki fragments are quite important to understand. And at the end of the chromosomes, you have telomeres that consist of that sequence TTA-GGG that are basically protecting the ends of the chromosomes. But what also happens is that with each cell division, usually these telomeres shorten. So every time you replicate them, they will get shorter and shorter. And that's one of the ways to tell how old the cell is, how many times it has divided, and often this will push the cell to go into senescence, so go into the state where you no longer divide. And cancers have developed multiple ways of dealing with that, which is really fascinating if you want to read about it. Okay, and of course, all of this process is quite error prone. So it's not as error prone in bacteria that make a lot of errors during the replication, but still we make, I forgot now about the frequency, but we make quite a few mistakes during the replication. But there are quite a few different enzymes that try to counteract it, and the polymerase itself has this proofreading capacity where it checks if the nucleotide that inserted is actually correct and is matching what it's supposed to be. And then if there is a break, there are two main mechanisms for sticking the DNA together. So if you get a double-stranded break where both strands break, there is a hole. There are proteins that lead to non-homologous end joining, which is one mechanism that basically puts a glue between this DNA and joins it together, leading often to mutations. Or homologous recombination, where you look for the other copy of the chromosome, and based on the template, you try to repair this DNA. So here, just for the interest of time, I'm not going to talk about it more, but it's something important to be aware of because many cancers lack one or the other of these methods, and it's very important in how we design the therapies to target them then. Okay, so yeah, a couple more MCQs before we finish, basically. So another 30 seconds for this one. Yeah, try to match the description on the left with option on the right. Yeah, so it should be quite easy. So DNA strands run in opposite directions, which means they are anti-parallel. Second one, the structure of double-stranded DNA provides an efficient framework for replication because, yeah, you've got the complementary strands. So I feel that's self-explanatory. How many hydrogen bonds will the sequence form with its complement? So that's more tricky one. If you remember, I don't know, it doesn't work for everyone, but for me, like this G and C seem to be like really thick letters. So a lot bigger than A and T, and they take a lot of space. So this is how I kind of remember that they form this triple bond. So for each C or G, 1, 2, 3, 4, you get 3 bonds. So that's 12 plus 1, 2, 3, 4, 5 times 2. So that's 10. So it will be 22 altogether. DNA replication is classified as semi-conservative, something we just talked about. One strand will always be always the old strand, the other will be always the newly synthesized one. And which of the following enzymes is involved in the process of DNA replication? Here it will be helicase that basically creates this wrap by just pulling this DNA together. Creates this DNA bubble. Brilliant. So we haven't really talked about transcription and DNA polymerases, but I feel unless you really want to, you can do it in your free time. But let me know if you have any questions or if you want to carry on or anything. But I appreciate it's really late and you must be dead. Yeah, if you have any questions, just put them on the chat or just speak up.